Continuing now with our chapter 9, talking about muscles, we're going to talk more about that muscle contraction and how it actually occurs. So the physiology, so we talked in the other video about the histology. So we have our sarcomere with our actin and myosin filaments, and we talked about the sliding filaments that allows the sarcomeres to shorten to cause contraction. All right, so now we're going to talk about the physiology, what actually makes all that happen. So as we mentioned, skeletal muscle is voluntarily controlled by our nervous system. So that controls the muscle contraction through those action potentials. So the motor neuron comes down, releases acetylcholine that acts on that motor plate to cause muscle contraction. So how does that really all happen? So first of all, we have a resting membrane potential. So potential just means opposites. Membrane, here we're talking about the voltage across the plasma membrane, or that sarcolemia, specifically in the muscle cells. So again, at rest, so when it's not being activated, the inside of cells is more negative compared to the outside of the cell. And it's more negative there mainly because of DNA and large proteins that have a negative charge to them. Now there's more potassium on the inside of the cell than on the outside. And so potassium kind of leaks out, not completely though, but it leaks out because the inside is a little bit more negative, the outside being more positive. But we get a little bit of leakage of the potassium out because there's a high concentration on the inside, low on the outside. The outside though is more positive, and there is more sodium on the outside. So we have more potassium inside, more sodium outside. Inside of the cell, more negative than the outside of the cell. Now, what maintains and keeps more potassium inside, more sodium outside is what's called the sodium-potassium pump. And that's going to help restore that membrane potential after a contraction. So this has to occur in order for the action potential. So the inside has to be more negative, the outside has to be more positive. Again, sodium needs to be higher outside, potassium needs to be higher inside, or we can't get an action potential. So a little bit of review here about membranes. So membranes are made up of a phospholipid bilayer. So just to draw them out, we have the phospholipid head, 2 hydrophobic tails, and then we have another layer of phospholipid that's essentially upside down. So again, hydrophilic head, 2 hydrophobic tails, and we form this bilayer. We have these 2 layers of phospholipids with their heads on the outside and the tails coming together on the inside to form that hydrophobic barrier. All right, so that's going to inhibit the movement of anything charged. So any of our ions or anything like that, they are not going to be able to get across this hydrophobic barrier here. So they're going to be inhibited by that hydrophobic region. So transport proteins are needed then, so they get inserted. Sorry about that. So they get inserted into this membrane, and what these transport proteins do is they're going to allow passage of molecules in and out of the cell that are charged or too big or polar that can't get across this hydrophobic region here. So we put the transport protein in it to allow them to move. All right, so then a little bit about ion channels. We've mentioned ligand-gated ion channels whenever we were talking about that motor end plate. So with these, a ligand like acetylcholine binds to the receptor, and whenever this receptor, whenever it binds to the receptor, so again, like neurotransmitter or acetylcholine, that acts like a gate. So typically this ion channel would be closed, but whenever a ligand binds to it, called a receptor, it receives a signal. Whenever it binds to it, it attaches to it. So like when acetylcholine attaches, a sodium ion channel opens. Okay, so typically the gate's closed, but if acetylcholine attaches, that opens the gate and allows sodium to move into the cell. Again, that's down the concentration gradient because we have a lot of sodium outside, not a lot inside. So sodium comes through that channel, but only if the gate opens, and that only happens when acetylcholine is bound. Now, once sodium comes in, we also have what are called voltage-gated ion channels. And the gate on these open when there's a change in that membrane potential. Okay, so remember at rest, it's negative on the inside, positive on the outside. But as sodium comes into the cell, it's positively charged. So that makes the inside of the cell less negative because it's more positive coming in. So that changes that resting membrane potential. And that change then can open what are called voltage-gated channels. They respond to a change in membrane potential. So they're closed unless the membrane potential closes, or unless the membrane potential changes, then that opens those channels and lets ions through. All right, so looking at this, actually being able to see it. So again, at rest, the inside of the cell is going to be more negative than the outside of the cell. To orient ourselves, this is the outside of the cell. This is the inside of our muscle cell. So again, the inside of the cell is going to be more negative. The outside is going to be more positive. These are not very pretty plus signs. Sorry about that. So again, the outside more positive, the inside is more negative. Okay. In addition, we have more sodium shown here as pink on the outside. We have more potassium shown here as purple on the inside. And then again, the inside is more negative because of all these proteins and DNA and those type of things that make it more negative on the outside. All right. So then we also have, again, potassium on what are called leak channels. So again, potassium moves a little bit at rest here. So again, these leak channels that are here shown in purple. So a little bit of potassium is going to leak out at rest, but not a lot because it's positively charged. And again, the outside of the cell is positive, so that kind of restricts the movement very much. But then again, we have our sodium potassium pump that's here as well. Again, pump indicates it needs energy or that comes from ATP. But again, what it's going to do is restore that. So it's going to send sodium out, and it's going to bring that potassium that leaked out or that came out back into the cell. So that keeps everything normal at rest. Again, positive outside, negative inside, higher concentration of sodium outside, higher amount of potassium inside. So then the action potential, a little background about those. What happens, we have a depolarization and a repolarization phase. So what's going to happen, again, at rest, we have a negative membrane potential because the inside of the cell is more negative than the outside. If there's some type of action, like acetylcholine binding to a sodium channel, now sodium is going to come into the cell. And as sodium comes in, that makes the inside of the cell less negative, okay, or making it more positive. So our membrane potential goes from something like negative 85 to maybe negative 50 or something like that. So if it reaches what's called a threshold, so it's not just a minor little change like from going from negative 85 to negative 80. It has to be a significant change in that membrane potential. Again, a lot of sodium coming in. That is going, if it reaches the threshold, essentially what's going to happen at this point at the threshold is this is going to open those voltage-gated sodium channels. So again, if acetylcholine initially binds, let me get rid of some of this. So whenever acetylcholine binds to those voltage-gated sodium channels, sodium is going to come in here. This is the depolarization as sodium's coming in. So as the sodium comes in, the inside of the cell becomes less negative. We reach our threshold here, and then what that does is it opens these voltage-gated sodium channels. Now more sodium is coming into the cell, and we get this depolarization where we actually switch from a negative membrane potential to a positive membrane potential. Now, what's going to happen at that point is that whenever the membrane potential becomes positive instead of negative, that opens voltage-gated potassium channels. Whenever those channels open, now the outside of the cell, let me just kind of draw this, whenever there's all this extra sodium on the inside, so we'll draw sodium here as red, now what's going to happen is the potassium is going to want to go outside from a high to low concentration like it did with the leak channels, but now because the outside of the cell is positive instead of negative like it was at rest, now there's a huge driving force for that potassium to come out of the cell. And as potassium comes out of the cell, now it's going to repolarize it. Now as that positive ion moves out of the cell, now our membrane potential starts to go back to negative. Once it reaches that, now it's repolarized. It's back to the normal resting membrane potential, where it's more negative inside, more positive outside. All right, so now what we're going to see is as it becomes again more positive, again, so as those potassiums come out, now it's more positive than it was negative. So now though, the problem is that our membrane potential is restored. It's positive outside, negative inside, but we have all this sodium on the inside and we have all the potassium that just came out. It's on the outside. So we need to get those two switched. And that's where that sodium potassium pump is going to come into play. So that's going to move that potassium back in and brings the sodium back out. Now it's repolarized. Now it's ready for another action potential. All right, so looking at these a little bit more. So our voltage-gated, again, ion channels. So again, at rest, the voltage-gated ion channels, which are shown here in red, one here, these are closed, okay? Because again, there's no change in the membrane potential, so that gate is closed. So sodium cannot move out of those cells. Or the sodium cannot come into the cell, sorry. Also, again, our potassium channels, which are shown here in purple, now they're open at rest. These are just called potassium leak channels. And again, a little bit of potassium is going to move through here, but not a lot, because while it's going from a high to low concentration, remember the outside is more positive, so that positives don't like each other. So that positive potassium doesn't move a whole lot because it's already a positive environment. So again, ultimately, this makes the inside of the cell more negative than the outside. So looking here at our membrane potential, that difference in charge across the membrane. All right, so now what happens during that depolarization is those sodium channels are going to open first because of the ligand gated. So again, acetylcholine is going to bind to those voltage gated sodium channels. And now, so when acetylcholine binds here, now the gate opens. Let me use a different color. So now our gate is open and now sodium is going to rush into the cell. Again, it's going from a high to low concentration. It's also negative on the inside at this point. So there's a huge driving force for sodium to come in, and it depolarizes the membrane potential, meaning it switches it. Now the inside is more positive and the outside is more negative. So it's switched, and that's due to that sodium coming in. Now, as the membrane potential changes, that's also going to open other sodium channels that are regulated by voltage-gated channels. So again, the acetylcholine starts it by binding to the voltage. The acetylcholine starts it by binding to those ligand-gated channels. But then as that sodium comes in and our membrane potential changes, now we've opened the voltage-gated channels. Now at a certain point then, whenever everything flip-flops, and now again the inside is more positive, the outside is more negative, what that does is it's going to open voltage-gated potassium channels. So here again our gate was closed that we see up here during depolarization, but when the voltage changes, when the membrane changes and it becomes more positive inside, now this gate is going to open, and now potassium wants to go out of the cell because it's going from a high to low the potassium, so now that potassium is going to want to go out of the cell because there's a lot of potassium inside of the cell here, these little purple circles. And where it is at rest, the leak channels didn't let much go through because the outside was negative or the outside was positive. Now that sodium really wants to go out of the cell because it's going from a high to low concentration, but also because the outside is now more negative. So now sodium rushes out, or potassium rushes out of the cell. And as it comes out, that makes the outside more positive and the inside more negative, and we restore that negative membrane potential. So that's referred to as repolarization. So if we summarize again what's happening here with our action potential, so at rest, again, a negative membrane potential. The outside of the cell is more positive. We have more sodium going on out here. The inside, we have more potassium, and it's more negative on the inside. All of the voltage gated, whether it's sodium or potassium, all those gates are closed that you see here, but we do have some potassium leak channels that are open, and a little bit of potassium can move out of the cell. But those voltage gated potassium channels, they're closed. So that's negative. Now, acetylcholine over here is going to get released. It's going to bind to these voltage gated sodium channels that we see here. Whenever acetylcholine binds, now sodium is going to come into the cell. And as sodium comes into the cell, it's going to make it change the membrane potential. It's going to be less negative initially, and then what that's going to do, it's going to open the gates of those voltage gated sodium channels. So when we reach that threshold, now the voltage gated sodium channels open and a lot more sodium comes into the cell. That's actually going to make the inside of the cell positive at this point instead of negative. That is then going to open up the gates of the voltage gated sodium channels. As sodium leaves the cell, that makes the inside more negative, the outside more positive, and eventually we get back to that negative membrane potential. Again, the problem at this point though, our membrane potential is negative again, but we have too much potassium outside and too much sodium inside. So now we need that sodium potassium pump to put those back in the right concentration. All right, so as we watch this action potential move down kind of the neuron, as it moves, it only goes in one direction. So again, this is our resting, so it's negative, outside is positive, but as that action potential, as the depolarization occurs, what's going to happen is now on this region, the inside is more positive because all that sodium came in, the outside's more negative. So that's going to open voltage gated sodium channels in this area that causes more sodium to come in. So now this region becomes positive on the inside, negative on the outside. That opens voltage gated sodium channels here, and it just keeps moving on down, kind of jumping a little bit as we go, as that action potential moves or propagates down that neuron. So now at the neuromuscular junction, again, there's a lot of information on this slide, but essentially what's going to happen, so our action potential is moving down through here, it's changing that membrane potential as it goes. Well, what happens whenever it gets to the terminal, to the end of it, it is going to open these voltage gated calcium channels. So voltage gated calcium, whenever they open, calcium is going to come into the neuron, and it's the calcium coming in that causes the synaptic vesicles to fuse with the membrane, and that releases the acetylcholine into that synaptic cleft, or that synapse, or the neuromuscular junction, the space in between the neuron and the motor unit. Again, that acetylcholine is then going to bind to these ligand-gated sodium channels. So again, they're closed unless acetylcholine binds to them. So when acetylcholine binds to them, they open, and now sodium is going to come into this muscle cell. As sodium comes in, it changes the membrane potential. That opens the voltage-gated sodium channels, and that's going to let more sodium come in. Now, what happens after that? So again, acetylcholine has bound. There's an enzyme called acetylcholine esterase that breaks down the acetylcholine. So this doesn't last very long. So it's a very quick response. Also, that acetylcholine can be reabsorbed. It can go back into that presynaptic neuron and get recycled. So that recycling, though, takes less energy than breaking it down. So it's more effective to recycle it. So then it can use it again if another action potential comes to that axon terminal. All right, so now what effect, though, do we get here with this excitation-contraction coupling? So this is going to link that electrical and mechanical components of our contraction. So essentially what's going to happen is that action potential produced by the sarcolemia. So again, that sodium rushes into the muscle cell, so into the cytoplasm of the muscle called the sarcolemma. So as sodium rushes into that, that gets propagated to those T tubules that we talked about, that transverse tubules. Well, what that's going to do is open calcium channels on that sarcoplasmic reticulum. So again, they're voltage gated. So just like with our neuron, when calcium rushes in, it releases the acetylcholine from the neuron. Well, on the muscle side, when sodium comes in, when acetylcholine binds to the ligand-gated sodium channels, sodium rushes into the muscle cell. That changes the membrane potential of the muscle cell. That opens voltage-gated calcium channels on the sarcoplasmic reticulum. Calcium rushes into the sarcoplasm, and that's what's going to lead to the muscle contraction, because we get binding of the troponin, and that activates the shortening of that sarcomere. So again, this action potential and the muscle contraction. So again, we have this acetylcholine. It's going to bind to those ligand-gated ion channels. So we have the ligand-gated sodium channels. So if acetylcholine binds, it opens the sodium channel. Sodium comes into our muscle. That changes the membrane potential of our muscle cell, that sarcolemia. And as that happens, calcium is going to move out of the sarcolemia due to that voltage-gated calcium channels opening. As calcium comes into the sarcolemia from those T tubules, what's going to happen is calcium is going to bind to the troponin. Troponin is then going to cause that cross-bridging of the myosin to the actin, and it's literally going to pull them together and allow that sliding to occur and that shortening of the sarcomere. All right, so just kind of looking at this cross bridge. So again, exposure of the active site. So this is kind of at rest, but now the calcium is going to attach to the troponin. So whenever that happens, now the myosin attaches to form that cross bridge. We get what's called a power stroke. So we get some ATP released here, and that head region of the myosin is literally going to power or move that. So as we get energy released here in the hydrolysis, now it lets go. That causes the actin, the myosin, to release the actin. Whenever it releases, it goes back to normal. So it lengthens again. All right, so just kind of looking at this in a little bit more. So again, that myosin head stores the energy from ATP breakdown. So the myosin head remains in the high energy position, okay, until the muscle fiber is stimulated by the motor neuron, and that causes the excitation contraction coupling to occur. So again, we have our active sites here on the actin where it wants to bind to the myosin head region. Again, we have calcium interacting with the troponin that alters that tropomyosin. So then as the cross bridge forms, so once calcium comes in and calcium binds to the troponin on the active sites that are exposed, the myosin head quickly attaches to them. So the influx of calcium causes the myosin head to attach to the active site on the actin. Okay, the binding of the myosin head to the active site forms that cross bridge, and that triggers the movement of the myosin head at this hinge. So remember, we talked about the hinge region, and then the movement of the head there is called the power stroke. So because the head is bound to that G-actin, the actin is pulled past the myosin, so that sliding filament that we talked about. So it slides past it toward the H zone. So our H zone would be in here where they're not overlapping. So during this power stroke, it pulls the actin across, it slides it across so that we remove that H zone. Okay, now binding of ATP to the myosin head causes it to detach. So ATP binds, it releases it from that active site, and then that myosin head breaks down the ATP that remains attached to the head. Okay, now the breakdown of ATP by the myosin, this leads to the recovery stroke. So this returns the myosin head to its high energy normal position, which is where it is not attached to those active sites. Now that it's no longer attached, everything goes back to its normal state. All right, so during a single contraction, each actin and myosin goes through a cycle of cross bridging. So it just was showing one at a time, but each of the active sites on the actin would be attached to a myosin. So it's a collaborative pull that shortens each of those sarcomeres. All right, so our summary here of our skeletal muscle contraction. So again, we have our neuromuscular junction. We have the action potential coming down. We open those sodium, or we open the calcium voltage gated channels. Calcium comes into our presynaptic cleft, causes acetylcholine vesicles to fuse with the membrane. We release acetylcholine. It binds to the acetylcholine gated sodium channels. That causes sodium to rush in. That causes a depolarization that causes calcium to be released from those T tubules into the sarcolemia. The release of calcium, the calcium is going to bind to the troponin. Whenever that happens, now the myosin is going to bind to the active site on the actin. That energy is used to go through that power stroke, so it slides the actin across to form the shortening of the sarcomere and that contraction. ATP is then added and everything goes back to its original state. All right, again, just a summary of each of those. I'm not going to walk through these. Again, I don't like a lot of words on the PowerPoints, but it is good to be able to walk through each step. So again, I've kind of talked you through it, but if you needed to walk through each steps kind of on your own, this is a nice summary slide to do that. Now the relaxation part again, so those three major ATP dependent events that are needed for relaxation. So after the action potentials occurred, those sodium potassium pumps need to restore the normal thing. So they need to transport sodium out and potassium in to the cell to maintain a normal resting membrane potential. So we need energy for that. We need energy for the sodium potassium pump. We also need ATP to detach the myosin head from the active sites for that recovery stroke. And then finally, we need ATP for active transport of calcium into the sarcolemma. So it's going to recycle the calcium that was released. It's going to put that calcium back into the sarcolemma so that it can be stored for the next contraction. Excuse me.